Mega is a freely available piece of software. It was developed with a grant, and there are continue to be updates. It's a very flexible and somewhat powerful piece of software. So rather than having you learn many different programs, we're going to try and do most, or even depending on the class you're in, all of our analyses using Mega. To download Mega, you go to megasoftware.net. It will come up here as the first window. Select your preferred operating system, either Windows or Mac. If you use Linux, more power to you, but you're probably going to want one of these two. Windows is what I'm going to be doing everything in. The Mac version sometimes is a little less stable, depending on your operating system. So I would recommend saving your work frequently if you're working in Mac. It's a good idea in Windows also, so you don't do a lot of work and then lose it. So once you hit download, it's going to ask you for just some very basic information. So you just go through and say accept, and then essentially you just can check one of these. I'm at home, so it says I'm under Time Warner Cable, but fill out that information and download it, and it's yours to use forever. Okay, so I'm going to demonstrate. I've already got it downloaded and installed. I'm going to do a quick run through and demonstration just so you can get familiar with it. I recommend that you pause the video here, download it and install it, and then follow along with me. You can put the video side by side or pause it and go back and forth. It will be helpful and help you to learn it if you are following along and doing it yourself. Now, there are all sorts of good tools that you have here, some of which we'll be using depending on the course you're enrolled in. But today we're just going to show you about alignments and how to build a data set, how to generate a data set. So I'm going to click on Align, Edit, Build Alignment, and then create a brand new alignment. And when I do this, it's going to ask me if I'm going to use DNA, protein, or I guess you can go back. But we're always going to select DNA. If you have a DNA alignment, you can always translate it and look at the protein view also. But if you have protein, you can only see the protein. So for all of our purposes, we're going to be using DNA alignments. Now, this is something that some people don't recognize, and it can throw them a little bit. You have a main window here, and then as you open up an alignment or an analysis or any of these other options, you're going to see additional windows that come up. It's not all just in one window. So the default here is just a blank kind of placeholder sequence with no data at all. And we need to begin to add data. Now, for your various worksheets and assignments, you might have different tasks that I'm going to ask you to do. If you are in the 6330 course, you're going to be asked to put together your own data set. So you need to select a gene, and you need to select a group of organisms that you're going to work on. I'm going to ask you to download at least 20 sequences. So let's look at how you would do that. So there are a number of shortcuts up here, or you can do the drop-down menu. So if you want to search for, under the web, I guess it is. If you want to find them, there are two ways that you can find sequences. The first one we're going to do is simply query GenBank. So this automatically, it opens up its own little separate browser window. You can use any browser that it takes you directly to NCBI, but the advantage of the Mega browser is it has this tool on it. You can add your sequences directly to your alignment rather than having to add the sequences manually and cutting and pasting. So by default, we're looking up at nucleotides, and let's just pick a gene. Now again, my hope is that if you are required to put together your own data set, you'll choose a gene that you're interested in and that has maybe even some relevance for the research that you're doing. If not, just pick a gene that you're interested in. So let's choose an opsin. Okay, opsin is a type of a gene that is involved in light perceptions, and different organisms have different types of opsins. So let's say we wanted to study the evolution of opsin number 1 across a group of mammals. And again, depending on your interest, you might choose a different gene, or you might choose a smaller or a wider range of organisms to study. So here it comes up as a gene, opsin number 1, and then below it are also many, many sequences. this is the chromosome that has the gene on it, but it's the entire chromosome, 156 million base pairs. And so you don't want to have to sort through all that to find the one you want. And then sometimes if you're just searching by keyword, you're going to have a bunch of other things here. But notice the very first one is that gene that we selected for. So let's go ahead and click on the link for that gene. And because you're now under gene, you have a whole bunch of information about the gene. You have information about where it's located, right? How many exons there are, a little graphical representation of where it's found on the X chromosome. This is a sex-linked gene. It's found on the X chromosome, which is one reason why males are more susceptible to color blindness. And then it's got tons of information about lots of research and other things that other ways it might be involved and where it might be expressed and have other functions. But for our purposes, we just want to find the sequence. So we're going to scan down until we find the mRNA. So here we are, mRNA and proteins. And we're going to click the link here for the first one. And sometimes, though, if there are variants of different alleles, you might have multiple versions. But for our purposes, it doesn't really matter which version you get. Okay, so Homo sapiens, opsin 1. Now, again, you've got a lot of information here, but at the bottom of this page, you have the DNA sequence. There it is. And so here's the advantage of Mega. When you click the Add to Alignment, the default is to import only the CDS, which is only the coding sequence, if it's available. If you have a sequence where the coding sequence is unknown or hasn't been marked, you'll have to import the full sequence. But for the vast majority, you'll have coding sequence that's been marked. So I'm going to open this up. It will automatically give it the name. You can change it if you want, or you can leave it the same. So I'll just maybe shorten this just for simplicity's sake and leave it as, that's the full information. Let's change the name here. Homo sapiens, opsin 1. Okay. When I do that, it's going to automatically load it into my alignment. My placeholder one's still there, so I can get rid of that one now if I want to. I'll just delete that. And here we have start codon all the way through. This one is, you click on the end, it'll tell you, is 1,095 base pairs long. There's our stop sequence, TGA. So that is just the coding region. We've automatically sliced out all of the introns. We have only our exons from start to stop codon all spliced together. So this is what the mRNA would look like, except you'd have U in place of T. Okay, so it's really nice and handy. Now, you can also translate this into the protein sequence. When you do this, it's going to ask you by default what the genetic code is. Now, since I am in a vertebrate chromosomal gene, I would just say yes, standard genetic code. But notice that if you say no, it's going to bring up other options. So maybe you are working with mitochondrial genes in vertebrates. And there are very slight differences to the DNA code depending. Most of these are very, very specialized. But standard will work if it's if it's on a chromosome and it codes for a protein, then standard will be the one you're wanting to choose, unless it is a bacteria. Then you have a slight difference in the prokaryote. So there we are. And you can scan through this. You can go back and forth. It helps to have something of a larger screen. Okay, but you can see the sequence, and then you can toggle back and forth now between DNA view and protein sequence. If I click that, then from now on I can just toggle back and forth. Now, only changes when you have a DNA alignment, you're only going to be able to make changes if you need to when you are in the DNA view, but you can go back and forth. So we now have one opsin. Now let's say that we wanted to add this same opsin, but from a different species. So we can search for it by keyword search. So let's do that. I can come back to my original, there we go, where am I? Oh, there we are. So I can come back to my original one and I can go back or open a new page. Let's just go back to, again, my original search page. Went too far. And let's look for a different species. So keywords can be useful sometimes, but also they can be a little bit difficult sometimes. So let's say I want to find opsin 1 in the gorilla. Okay, so it's searching for it. There we go. And again, look at it carefully. This is an entire genome, so it's maybe there, but that's a huge. So again, with keywords, sometimes it can be tricky. So all I'm getting are genome results, 152, 127 million base pairs. I'm not certain that it's there, and if I did, it would take a long time. But as I get down, oh, look, here's one, opsin 3. It's not quite the one I want, but we're getting closer. And sometimes, depending on taxonomy, names of the genes, you might not know for sure if it's the one you need or not. So this can be a little bit tricky. Uh, if a gene has an abbreviated name, that can be actually helpful. So the abbreviation for this one is OPN, opsin 1. And let's see, I think that there's no space between the 1 and the end of it. Let's see, where's the abbreviation for 3 there? Yeah, OPN, OP5. So let's do opsin 1. And let's do a different search. So keyword search can be useful, but they can also be a little bit problematic. So I'm going to show you an alternative way if we can't find gorilla opsin here. Okay, no items found. That doesn't mean it's not there. It just might mean my keywords aren't bringing it up. So another option, if you come back to your alignment, under web, the second one is do a blast search. And a blast search is like a DNA search for, or like kind of like a Google search, but for DNA. Now, because I was in the protein view, it's going to look for proteins. I want to go back to my DNA view. I can even pick a single one like Homo sapiens, and there's a shortcut here. It's this one right here. That little icon is for blasting whatever sequences you've selected. And I want to find, let's say I don't care if specifically that's a gorilla, but I want to add an additional primate sequence. So I could come into this one and say, okay, blast search that, but use primates. Okay, so you have to wait for it to load up here. Try again, primates. If I search only this one, I'm going to get, because there's been so much human sequence that's been sequenced, I'm going to get lots of really close human matches. But let's go ahead and do it. Okay, so without limiting the organisms, I'm just going to do a quick blast search for this. And it's going to submit it. Sometimes it can take up to a minute or two, but usually it's rather quick. And it refreshes every few seconds. So we'll see what an unconstrained search yields if we're looking for a match for opsin. So once it completes, the result window will look like this, and you'll have a list of all of the DNA sequences that produce significant matches and how close they are. The lower the E value is, the better the match it is, and then how much of it overlaps. So notice, of course, all of our matches are the gene that we're searching with, Homo sapiens 1. But look, here's our gorilla, which is very, very closely related. 97.81% identity, very, very high E value. So let's bring that one in. And again, if it was a gene that I was unfamiliar with or a gene family like this one, I may want to be a little bit careful and do a little bit of research to make sure I've got the actual gene that I'm looking for. But for most genes, you'll be fine using either of these methods, searching for the gene name or searching using a blast search and finding the species that you want. And you can use the taxonomy tool to limit or extend your searches to specific groups of organisms. Okay, we'll pull in one more here in a little bit. So here it is. It's now loaded. I'm going to add this one to my alignment, only the coding sequence, and push OK. We'll just leave the name the default. Now, if it says predicted, that simply means that they haven't verified using other methods other than the DNA sequence what gene it is. But predicted genes are just as good as ones that don't say predicted. Okay, but just so I can see the name of the species first, I'm going to change that. I could have done that originally from the earlier portion also. Okay, gorilla, gorilla, long wavelength. Okay, now notice that it looks quite different, that we have different sequences here. Okay, and mostly that's because we have not yet aligned these genes. Let's bring in one more. I'll bring in a chimpanzee. In fact, if I come back here and let's close that window and go back to my matches, which are up here at the top. Oops, descriptions. Okay, I have another match that's very, very close. That's right here. That is opsin 1. I don't recognize the genus and species, but let's download it and see what it says it is. And there's Pan-Troglodytes, which I know is a chimpanzee. So we'll do those two more, and then we'll show you alignment. So here it is. There's the matches that it's making. I'm going to click on the link to the sequence. Try that again. There's the link to the sequence. I'm just having connection issues. Okay, here we go. And it'll tell you what it is. So this is a gibbon, one of the closely related to the hominids, but not part of the hominid family. It's in the Hylobatidae family. So we can look at the sequence if we need to, or simply just add it to our alignment. Let's do that one. We'll change the name. So species is first. I don't know why. I guess must have not changed it before I hit enter, but okay. There we go. Now let's add that one last one. So I'm going to close this tab, come back to our descriptions, and let's add the chimpanzee. There's our link. Here's our sequence, add it to our alignment. And so now I have 4 different DNA sequences, and notice that the alignments don't match exactly. Now, when you are using proteins, usually the best way to do it is to translate them first. I want to make sure that I am completely in the right reading frame, that I don't have any stop codons. I don't have any stop codons till the end. And then I'm going to align them. You have 2 options for aligning. And we'll talk about alignment later, but we'll use clustal, which is that little W. You could also use muscle. Either one will be fine. Align them at the protein view. Oh, and I need to select them. So we'll select all of them. And then I'm going to align them at the protein level. You can leave all of the search parameters at default, or the alignment parameters at default, and push OK. So now notice at the protein view, we have quite a bit of sequence conservation. There are differences here and there, right? So we have some differences. And then there's a little difference in length. So the gorilla and the chimpanzee have a longer sequence. It looks like there has been a deletion in one or more of these lineages. And we'd have to look at a phylogeny and map them on, which is stuff for later in the semester. Okay, but notice there are polymorphisms. So there are sites where amino acids are variable. And if we look at the DNA sequence, we're going to see even more polymorphisms with more changes at the DNA level. Not all changes at the DNA level lead to a change at the amino acid level. So for instance, here we have one that I would bet that when we translate it, yep, there it is, everything is the same, okay? So this is how you're going to build a data set. And again, you might need to do this, or you might be given data sets depending on which class you are in. If you still have trouble with Mega or are needing some help, email your instructor or log on to one of the Zoom sessions and we can review it and I can go over whatever troubles you're having.